Welcome back. In the previous session, we have discussed about taxonomy, which includes the identification, classification, and nomenclature of organisms. In this session, we begin with the new lesson, that is biological classification. Now, as you already know, classification is grouping of all the organisms present into various categories based on their similarities and their differences. Studying of classification is very important because number one, it helps us to study the organisms in an easy manner. Second, we need not study all the organisms. If you study one or two specimens belonging to a particular group, it is like studying the entire group. When we classify organisms, we can also compare the properties of organisms belonging to a particular group. Not only that, we can compare the properties of different organisms of different groups. Not only that, classification also tells us about the evolutionary history of the organisms, how organisms have developed and evolved over the years. The first attempt of classifying organisms was done by Aristotle. Aristotle classified all the organisms into plants and animals. Very simple classification. He did so based on their morphological features, means how do they appear on the outside, morphology, and based on their morphological features, plants were further classified into trees, herbs, and shrubs. That is very simple. And animals were classified based on whether they have red blood or white blood. Later, Carolus Linnaeus, so we classified them under kingdoms. So the plant kingdom and the animal kingdom. The problem with this two kingdom system of classification is that under kingdom Plantae, organisms ranging from bacteria, cyanobacteria, fungi, and also gymnosperms, angiosperms, all these were put under the kingdom Plantae, which is not right, because we know that bacteria are completely different set of organisms, whereas algae, fungi, gymnosperms, angiosperms, they are completely different. This group included organisms which are unicellular as well as multicellular. So there was no differentiation made between unicellular organisms and multicellular organisms. Not only that, organisms that are autotrophic and organisms that are heterotrophic, both were combined in the same group, and there was no distinction made between the differences in the cell wall of the organisms. Let me give you a couple of examples. Take for example bacteria. Bacteria is unicellular. This is placed with a multicellular algae or a multicellular gymnosperm. Bacteria on one hand is a prokaryote. They do not contain nucleus and nuclear membrane is absent. This has been placed together with algae, fungi and plants which are eukaryotes. They have a nucleus and a well developed nuclear membrane is seen. So there was no distinction made between this. Take for example Chlamydomonas and Spirogyra. Chlamydomonas is unicellular, whereas Spirogyra is multicellular. Both are put together in kingdom Plantae. Not only that, fungus which are saprotrophic are placed with other plants which are autotrophic, means the mode of nutrition was not distinguished. And another case to study here is, even though there is a difference between the cell wall of fungus which is made up of chitin and the cell wall of plants which is made up of cellulose, this distinction was not taken into consideration. So the two kingdom system of classification of kingdom Plantae and Animalia had many demerits. It did not take into account the differences in their nuclear structure, whether organisms are prokaryotes or eukaryotes. Unicellular and multicellular organisms were not placed far apart. The mode of nutrition of organisms was not taken into consideration. Not only that, the mode of reproduction was not considered, and the cell wall differences was not considered. As years passed on, and with the advancement of science and technology, not only that, with the advancement of microscopy, more and more features were considered for grouping of organisms, and a advanced system of classification was then provided by R.H. Whittaker, which is called as the Five Kingdom System of Classification. Robert H. Whittaker, he considered the following features for arriving at this Five Kingdom System of Classification. The cell type, that is whether organisms are unicellular or multicellular. Not only that, he also looked at the genetic material, that is whether the genetic material is present inside the nucleus or it does not have a nucleus. That is, organisms were classified based on whether they are prokaryotes or eukaryotes. Then the composition of cell wall was also considered. He studied various groups of organisms and their cell wall composition, and this was also taken into consideration. The mode of nutrition, whether organisms are autotrophic or heterotrophic, or whether they belong to another group, that is symbiotic, all these were also considered. And sexual reproduction was also considered for classifying the organisms. So, based on these five features, Robert H. Whittaker, he classified all the organisms into Kingdom Manera, Protista, Fungi, Plantae, which we call it as plants, and Kingdom Animalia, that includes all the animals. This is the Five Kingdom System of Classification. With this Five Kingdom System of Classification, the demerits of the Two Kingdom System of Classification were mostly covered up. The Monerans, it includes all the unicellular organisms which are prokaryotes. So all the unicellular prokaryotes were included under Kingdom Manera. Protista includes unicellular eukaryotes. So these 2 points were used to classify them. And then the fungus, based on their cell wall structure and their mode of nutrition. Fungus has a cell wall made up of chitin and they are saprotrophic. They are not autotrophs. So fungus was removed from kingdom Plantae and placed in a separate group, which was not done in this type of classification. And plants included all the autotrophs. All the autotrophs comes under plants, and animals, that is without cell wall. So both fungus and plants have cell wall, whereas fungus are saprotrophs and plants are autotrophs. Animals don't have cell wall and they are heterotrophs. So these are the criteria for classifying the organisms into the 5 kingdom system of classification. This is what we will be studying in detail in the next 3 chapters, that is this chapter as well as in the next 2 chapters. In this chapter we will be studying in detail about kingdom Manera, kingdom Protista and kingdom Fungi. In the next lesson we will be talking about the plant kingdom, and in the 4th lesson we will be talking about the animal kingdom. So let us begin the discussion of kingdom Manera. Another important criteria that Robert H. Whittaker used other than this was that he also studied their evolutionary relationships. So evolutionary relationships were also considered when this system of classification was done. Basically, the phylogeny was also taken into account. How 2 groups of organisms are related to each other. So based on that also 5 kingdom system of classification was done. Let's begin with kingdom Manera. Kingdom Manera is also called as the bacterial family. It includes all the known bacteria in one kingdom, that is Manera. So let us write down the important features of kingdom Manera. Number 1, all organisms present in kingdom Manera, they are unicellular. They have a single cell only, and these are, meaning they do not have a well organized nucleus. Nucleus is absent. Nuclear membrane is also not present, it is absent, and they do not have any membrane bound organelles. Inside the cell there are many organelles like endoplasmic reticulum, Golgi apparatus, mitochondria, chloroplast, vacuole, etc. So all these membrane bound organelles are absent. So these are the prokaryotes, and the only organelle that they have is a 70S type of ribosome, which they use for manufacturing of proteins. So only ribosomes are present which are of 70S type. Talking about their cell structure, what does a bacteria look like? So based on the shapes, bacteria is classified into 4 types. Number 1 is coccus. Number 2, bacillus. 3, vibrio. And the fourth type is called as spirillum. Coccus are those bacteria that are spherical in shape. If there is only one single bacteria, it is called as micrococcus, only one. If there are two together, it is called diplococcus. If there are 4, tetracoccus. If these cells are arranged in the form of a chain, it is called as streptococcus. And then there is staphylococcus, which is a cluster of these spherical cells. So these are the spherical shaped bacteria. The bacillus is a rod shaped bacteria. The vibrium, which is singular, it is a kidney shaped or a comma shaped bacteria, and spirillum is a coiled bacteria, that is coiled. So these are the common shapes of different bacteria that are seen. Any bacteria, as you know, let us take the diagram, is a single cell. I am taking the example of a bacillus, a rod shaped bacteria, and it has a cell membrane. On the cell membrane there is a cell wall, and some bacteria, they have an extra outermost covering called as the capsule. So it has a capsule, the cell wall, and the innermost plasma membrane or cell membrane. At the end of the bacteria, at one end or on both the ends, they have a long whip-like structure called as the flagellum. This flagellum will help in motility of the bacteria. It will help in the movement of the bacteria. There could be one flagellum or there could be more than one flagellum. Other than the flagellum, there are tiny hair-like structures on the outer surface of some bacteria. This is called as the pili. P-I-L-I, pili, or singular is pilus. Pili are responsible for sexual reproduction in some bacteria. That is number 1. Not only that, pili help in attaching the bacterium to the surface. It could be a host or it could be any other surface for attachment and for sexual reproduction. Inside the plasma membrane, we find the cytoplasm. In the cytoplasm, we find ribosomes, which is of 70S type, and the genetic material is DNA. The DNA is present. There is no nucleus, there is no nuclear membrane. With the help of certain proteins, the DNA is present in a region called as the nucleoid region. So it is not spread everywhere in cytoplasm, in a small area called as the nucleoid. Other than the chromosomal DNA, bacteria also contain double-stranded circular DNA called as plasmids. This is a unique feature of bacteria. These are extra chromosomal circular DNA. Plasmids are known to offer antibiotic resistance in bacteria. That is the reason most of the antibiotics are ineffective against bacteria. Number 1 is they have got a tough cell wall and a capsule covering. Not only that, the plasmids contain certain antibiotic resistance genes which offer them resistance against the toxic antibiotics, which will help them to survive. That is the general structure of a bacterial cell. Bacteria are universal in occurrence. We find bacteria everywhere, in air, in water, in soil, in hot water springs, in deserts, in the cold mountain glaciers in the Arctic. Also, it is present on the body of animals, inside plants and animals. They are present everywhere. This is because of the tough cell wall and the capsule covering. Kingdom Manera is further classified into two types: Archaebacteria and Eubacteria. The cell wall is made up of peptidoglycans. In Archaebacteria, peptidoglycans are absent. Instead, they contain what is called as a pseudomurine. So, based on the chemical composition of cell wall, Kingdom Manera is further divided into Archaebacteria and Eubacteria. This is what we will discuss a little while from now. And the mode of nutrition: bacteria are autotrophs, they can prepare their own food. Also, they are heterotrophs, they are dependent on other organisms for their food. So, both autotrophs and heterotrophs are seen. And finally, the mode of reproduction. How do Kingdom Manera bacteria reproduce? How do they produce new offsprings? Mostly they follow asexual mode of reproduction, which is called as fission, wherein when the cell becomes mature, it just elongates and it breaks into two, producing two cells. That is what is called as fission. Sometimes, under unfavorable condition, bacteria also produce spores. Some bacteria are also known to undergo a special type of reproductive event called as conjugation. Conjugation. It can be called as sexual reproduction, wherein two bacteria, they come in close contact with each other, and through the plasma membrane or with the help of the pili, the genetic material is transferred from one cell into another cell. So there is an exchange of genetic material. It could be chromosomal DNA, it could also be plasmid DNA. So it is exchange. That's what is called as conjugation, come together. So this is a type of sexual reproduction. That is about the general properties of Kingdom Manera or the bacterial family. As the name indicates, archaic means very old, primitive organisms. These are the oldest organisms that we know. So that is placed under Archaebacteria. And I already told you, the cell wall of Archaebacteria is very thick and resistant. It is made up of pseudomurine, whereas here we have peptidoglycans. So based on that, this classification is then called as extremophiles. Phili means to love, extreme conditions. So these are organisms that are found in extreme conditions, such as in hot water springs where normally other organisms do not survive. They are found there, and in high salt content areas and in marshy conditions. So these are your thermoacidophiles. They survive in temperatures more than 100 degree Celsius, high temperature, and acidophiles, that is in high acidic conditions. These are called as halophiles. In salt concentration more than 30-40%, these bacteria can survive, and in marshy area, these are the methanogens. These methanogens are used to produce biogas from cow dung. Because they are present in such extreme conditions, these are called as extremophiles, and these are anaerobes, meaning they do not require oxygen at all. They can survive without oxygen also. So these are anaerobic bacteria. And archaebacteria, they are heterotrophic as well as autotrophs, both. Coming to eubacteria. Eubacteria, the word eu refers to true bacteria, in the sense that they have a peptidoglycan covering. This eubacteria, it includes your cyanobacteria, that is commonly called as blue-green algae, even though it is not algae. So that is cyanobacteria. They contain the pigment such as chlorophyll A and carotenoids and xanthophylls, which are seen in plants. So they can prepare their own food with the help of these pigments. So these are our autotrophic bacteria. Also are known to fix atmospheric nitrogen. They can convert the nitrogen gas into nitrates and nitrites with the help of specialized cells called as heterocysts. As you can see in the diagram of nostoc, there are specialized cells which can fix nitrogen. These cells are called as heterocysts. So these are autotrophs. Also these are chemoautotrophs. In autotrophs there are two types: photoautotrophs and chemoautotrophs. Ones that prepare their food using light are photoautotrophs. The others which use inorganic nutrients such as hydrogen sulfide, iron, sulfur, etc. are called as chemoautotrophs. So we find both kind of bacteria in this category. We have another group of organisms that are mycoplasma. These are the bacteria without cell wall. These are the smallest organisms that are known. Smallest organisms. Why? Because they don't have any cell wall, and these are non-pathogenic. They are not known to cause any diseases in plants or animals. So these are non-pathogenic. Now, and of course there are heterotrophs also here. Heterotrophs. because bacteria are present everywhere, the bacteria could be useful, they could be harmful. For example, certain pathogenic bacteria are known to cause diseases in both plants as well as animals. So these are the pathogenic bacteria. Other than that, bacteria have got several uses. Number one, because most bacteria are heterotrophs, meaning they are dependent on other organisms. Bacteria are one of the best decomposers in the environment. They break down the dead plants and animals into small organic matter. So they help in nutrient cycling. The nutrients present in the dead plants and animals with the help of bacteria such as heterotrophs can be once again taken back into the soil so that plants can use these nutrients. So not only that, many heterotrophic bacteria are used in industries. For example, converting milk into curd is one example which we use in our daily life. Some bacteria are also used to make antibiotics for medicine industry. Bacteria are used in biogas facilities. As I said, methanogens are used to convert the animal waste into biogas. In the field of food, medicine and in industry and also for nutrient recycling, bacteria play very important crucial roles. That is about the first kingdom, kingdom Manera. Next we will discuss about kingdom Protista and kingdom Fungi. Thank you.